All right, quick review. We've been talking about the central dogma of genetics the last 3 or 4 days. We know that every living thing starts out with its genetic material being in the form of DNA. What's the nickname for the DNA that our outline writer likes to call it? The $1 million blueprint. And it's a blueprint for what? Everything, right? It's a blueprint for you. It's a blueprint for life. And life is made out of what? Proteins. Okay? So it's a blueprint for life. It's a blueprint for proteins, because proteins make life, basically. So if you've got $1 million blueprint, though, you're not going to just take it out to a construction site. You're going to put it somewhere safe because it's valuable. You're going to make a cheaper copy of your blueprint, and in the body, that cheaper copy is what? RNA. In the process of using the DNA to make a more portable, cheaper copy called mRNA, that process is called what? Transcription. And that's what we concentrated on last class. That RNA has the ability then to leave where and go where? Leave the nucleus and go out to the construction site. And what is the construction site? The ribosomes. The ribosomes read the code, they read the blueprint, and then they put together what? What are the pieces? They put together the amino acids and end up with what? Proteins. And then proteins go on to determine your traits. So let's put protein right here. And the process from mRNA to protein is called translation. And then we know that proteins lead to traits. And then the one thing that we're leaving off over here is our little loop, and that little loop is called replication. And when did we say replication has to happen in a cell? Before what and what? Before mitosis and before meiosis. And the one exception to this, as far as we know, is what? Retrovirus, because they don't have any what? Don't have any DNA. The step they have to do first is called reverse transcription. All right, it's a good place to start with that. Let's go back and think about this transcription stuff for a minute. Reverse transcriptase or just transcriptase? Reverse transcriptase is the enzyme that takes the RNA of the virus and makes it into DNA. So it's the opposite of transcription. Transcription takes DNA and makes RNA. Reverse transcription takes RNA and uses it to make DNA. All right, so with transcription happens anytime you need a gene to be turned on, right? For a gene to be turned on, it has to be transcribed and then what? Translated. So we said before that if we had a gene, maybe up here is part of a chromosome, and then we've got a gene sequence. Okay, so there's one strand of the DNA, and I'm going to go ahead and separate it out so I don't have to do that later. Here's the other section of DNA. These sections are still connected up here at the top, and they're probably still connected down here at the bottom. But this code right here is a gene. One of these sections is going to run from 3 to 5, and the other is going to run from 5 to 3. Oh, yeah, I did, didn't I? Thank you. I said it right. I wrote it wrong. 3 to 5. If this top is 5, this top has to be 3. If this bottom is 3, this has to be 5. Thank you. All right, something's going on in your body that you need this particular protein. There's some reason your body needs to turn on this gene. So down here, just below the gene is what? What's down here at the bottom of the gene? Or we might say it's the front edge of the gene. What do we need to have down there? Yeah, we need something called the initiator site. Or initiation site. Sometimes that's called the Tata box. because that initiator site, the site that says, hey, here's the beginning of the gene, it's loaded with what? Amines and adenines, okay? And that basically says, start here. Here's the beginning of the gene. Start here. Start transcription here. And then guess what's at the end of the gene? What's the opposite of initiation?
Termination.
Termination, okay? So at the end of the gene, you've got a site that says, okay, this is the end of the gene, stop here. It's a termination site. In between the initiation site and the termination site, that's the good stuff. That's the gene. That's the code. So if we're going to do transcription, the main enzyme involved and the only enzyme I want you to know about transcription is called RNA polymerase. That enzyme comes in, it finds this initiator site, and causes the DNA to unwind. It unzips the DNA itself. Unlike replication, where both strands are used as a template for copying, in transcription, how many of the strands are used to make RNA? Just one. And we call that strand what? The template. The template strand of DNA is always going to run from what to what? It's going to run from 3 to 5. Because RNA polymerase has to build RNA which way? 5 to 3. So if it's going to build on to this strand, this 3' strand, what it's got to put right here is a 5' carbon, right? If this is 3, then this has to be 5. And it's going to build that new RNA from 5 up toward 3. It does that. Wherever it sees a T, what does it put? What kind of nucleotide does it put across from a T? An A. And then if it keeps going, it sees an A, what's it going to stick in there? It's going to stick a U. I hadn't done that yet. It's going to probably be 2 or 3 weeks. We'll go into the first period. Go into the first period teacher. So this is just me listening to the announcement from your first period teachers. All right, so when it sees an A, it's going to put in a U. Remember, it's putting in nucleotides, or the guy doing the outlines calls them nucleosides. Those are nucleotides that are ready to get put into the chain. A, and then he's going to put in a C, and a C, and a G, and a G, and an A, and a U. And eventually, RNA polymerase gets to this termination site and says, what? Stop. And the RNA breaks free. When the RNA breaks free, what happens to the DNA? It wires back together. If this is happening in a eukaryote, what has to happen after transcription in a eukaryote? In you, what happens after transcription? Before translation, what happens between transcription and translation? There's a step called processing. Okay? Exactly. So let's go to a slide and look at the way that works. But before we get to all that, though, let's remind ourselves of a couple things. Transcription happens in 3 steps. Initiation is the first step. And initiation is where the RNA polymerase II, that main enzyme, binds to that initiation site. Start here. Start here. That's the start here step. After initiation, we get elongation. What are we elongating? What are we making a long chain of? RNA, right? Nucleotides, RNA nucleotides. And that's always getting built from 5 to 3. The RNA polymerase II is adding in those nucleosides, making a long chain. And then the last step is termination. Eventually, the RNA polymerase gets to the termination site, the stop code, and it stops. And remember, once it stops, the RNA breaks free from the DNA. The DNA winds back up. After that, then if we're in a eukaryote, we've got to get some processing. If we're in a prokaryote, where does that RNA go? It goes straight from transcription to what? To the ribosomes, and it gets translated. No processing needed. But in a eukaryotic organism, we do quite a bit of modifying. What's the name for that original piece of RNA that gets made during transcription? The first piece of RNA we make right after transcription is finished, It's called the primary transcript. But that's not a finished product. We gotta improve on that zone. So in a eukaryote, the first piece of mRNA that's made is the primary transcript. Then we're going to do some add-ons to it and some takeaways. We're going to add something on the front called a 5' cap. And like it says here, it's sort of like putting a hard hat on. It's to protect it. It's also to help it to attach up to the ribosome so that it'll work correctly. On the back end, the 3' end, we're going to add what's called a tail, a poly-A tail. A lot of adenines. And this tail is about keeping your own cells from breaking down your RNA. It's to keep the RNA so that it'll hang around for a while, it can be used. And then other than that, we also do some middle modification. And this is where what gets taken out. The introns. Remember, some of your DNA, depending on the situations in your body, certain nucleotides don't actually code for anything. And those need to get removed before translation. Those pieces that don't code for anything are called what? Introns. The pieces that do code for something are called exons. So during processing, the introns get taken out and the exons get put back together. And the enzymes that do that are called spliceosomes, or sometimes they're referred to as SNRPs. SNRPS. Kind of the cool thing about this, though, is depending on what's going on in your body, the introns one time may not be the introns on that same gene the second time, which means one gene actually has the ability to do what? Code for more than one protein, depending on situations in your body. Sometimes we call that alternative splicing. What's taken out one time may not be the same thing that's taken out the next. And that lets your body respond to its environment and make different proteins when it needs different proteins. It gives your body the ability to make it on a few genes and still be able to make a whole lot of proteins. Each gene can code for multiple things. That's kind of important. All right, here we see some transcription going on. And we can see that only the template strand, also known as what? The sense strand. Only that strand is used to code for RNA. The RNA is getting built from 5 toward 3. You see this little bubble where it's happening. Sometimes that's called the transcription bubble. And the enzyme that's building the RNA is RNA polymerase. And it's always building it from 5 toward 3. And remember, this is happening where? Where in a cell does transcription have to happen? It has to happen in the nucleus, because that's where what is? For the DNA. Once it's finished, where's that RNA going to go? It's going to go to the ribosomes, which are where? They're outside the nucleus, out there in the cytosol or the cytoplasm, right? They're not in the nucleus. All right, let's talk about translation. Translation happens after processing in a eukaryote. Translation happens at the ribosome.
It has the same 3 steps as transcription.
Same 3 steps: initiation, elongation, and termination. And let's go to a slide where we can see some of that. Actually, before we get too much into it, let's talk a little bit about a ribosome. Remember, a ribosome is not considered an organelle. Prokaryotes and eukaryotes both have to have them. It's sort of a cell tool, we might think of it as. What they call a cell particle. These ribosomes come in 2 parts. And they're actually called parts that sort of make sense. The small part, the small subunit, and the large subunit. The small part is sort of like a table, a platform, where the work is done. And the large part is actually like the factory that'll set up on top of that table. So you got a table, and then you got the machine that sits on top of it. The large subunit has a couple of what are called sites inside it where certain things happen. The A site, actually let's start with, let's see, where should we start? Let's just start with the A site. The A site is where a new tRNA molecule will come in and attach. What do tRNAs carry around. I told you last time to think of them as taxis or what? Amino acids. So a new tRNA is going to park. It's going to park in the A site, and it's going to deliver its passenger, an amino acid. The P site is where these amino acids actually get hooked together, where one amino acid gets hooked to the next one. And then finally, the E site is like an exit site. Once it's finished, once everything's been attached, the used up tRNA, the taxi, he pulls out the party light. And what does he go look for? Another passenger, another amino acid. So think of the A site. This is where the taxi is going to park, and he's going to bring in his passenger, his amino acid. The P site is where his passenger meets up with whoever he's waiting on. And who is he waiting on? Another what? Another amino acid. They get hooked up. They grab each other's hands. They start holding hands. They attach. What do we call that attachment between two amino acids? A peptide bond. So they make a bond. They're holding hands. They're attached. And then the E site, once the taxi, the tRNA has let out his passenger, he's going to pull out and leave and go try to find another passenger. That's what happens at the E site. It's the exit. Big bold letters here. Remember, ribosomes are not organelles. Every cell has them, prokaryotic, eukaryotic. Think of them like cell tools. There's a picture of a ribosome in general. Here's the small part where the mRNA first attaches. And after it attaches, then the big subunit, the large subunit comes in and starts doing the work. Let's start here. We get initiation first. Notice in initiation, the small subunit of the ribosome first attaches to that 5' cap. Remember, that's what that cap is for, to help it to attach. Every mRNA starts with a codon called AUG. This is the first 3 letters in every mRNA. And this basically says what? What do you think AUG means? Start, initiate. It's called the initiator codon. Start. We're going to use that word codon a lot in translation. A codon refers to 3 base sequences found where? We used the word codon in conjunction with DNA back a little while ago. But when we're talking about translation, a codon refers to an A, T, G, or C combination of 3 letters found on an mRNA. When you hear codon, I want you to think mRNA. And I'll tell you why in a second. But right now, you hear that word codon, 3 letters on an mRNA. 3 nucleotides on an mRNA. After the small subunit gets connected up, the large subunit comes in and binds to it. Takes a little bit of energy for this to happen, and GTP provides that. Remember, GTP is very much like what? ATP. It's an energy molecule. The large subunit gets lined up so that the first amino acid can get brought in. And it turns out the first amino acid that gets put in the front of every protein is an amino acid called methionine. Because what does AUG code for? What does each codon code for? 1 amino acid. And it turns out AUG means that amino acid. And so it always gets put in front. Now, sometimes it gets removed later. The protein may get changed around a little bit later. But proteins always start with AUG at the beginning. It may get taken away later, but it starts out there. All right, let's go back a slide or 2. So here we see initiation. We see the small subunit of the ribosome attaching up to the mRNA. AUG gets in place. Also notice that a tRNA brings in this first amino acid called methionine. Now here's something we haven't mentioned. What do we say we call this AUG? Start what? Since it's on mRNA, it's a start codon. The tRNAs, the taxis, have to know where to park. They have to make sure they park in the right place. How do they know where to park? They have a code on them too. Every tRNA has a code on them too. And what's their code called? What's that UAC called? On an mRNA, the three-letter codes are called codons. On a tRNA, they're called what? They're called anti-codons. Anti-codons. What happens? The only way this amino acid can be parked in that spot is if what? If this code is complementary to this code. It can't park there if it doesn't have the right code, is what I'm saying. So that ensures that the right what gets where it needs to go. What do we say tRNAs carry? Amino acids. If the code don't fit, a protein you don't get, okay? The tRNAs and the mRNAs, they have to match up. If they don't, it doesn't go there. You get the wrong amino acid. So once the methionine is brought in by the tRNA, the large subunit of the ribosome comes in. Remember, the P site is sort of where it parks. Park there. And that's where we leave initiation. What comes after initiation? What came after initiation in transcription? Elongation. In transcription, we were trying to elongate the chain of what? mRNA nucleotides. In translation, we're trying to elongate a chain of what? Amino acids, so we can make a protein. So let's go to elongation. So in elongation, this is where the amino acid chain is actually made. What do you think that right there means, that 1 prime? What's 1 prime maybe stand for? What does it sort of sound like? Primary. Remember when we were talking way back to the structure of proteins? What was the primary structure of a protein? The order the amino acids are in, right? Which ones are there, and what order are they in? So that's what we're doing here. We're putting them together, the right amino acids in the right order. We're building the primary structure of the protein. Exactly. We got to build the primary first. That's what we're doing. We're building the primary structure. To do that, the ribosome basically walks down the mRNA. 1 codon at a time, using GTP as energy, and it starts putting in the right amino acids, attaching up amino acids to each other. Yeah, the ribosomes walk down the mRNA, reading the code, putting the right tRNA in place, and if the right tRNA is in place, what's on that tRNA? What's it delivering? What a tRNA packs in? Amino acids. They put the right one in place. GTP is also used to make the bind between the 2 amino acids. Takes energy to make binds, right? Binds are making of binds. Is that an endergonic or an exergonic process? Endergonic, which means you got to do what? Add some energy to make it happen. Yeah.
So the ribosome basically tells the tRNA, the amino acids where they're going, like traffic directors kind of
Sort of, yep, sort of like that. So I want to go to a picture here, and maybe I can explain it better from the picture. All right, here we see elongation. Remember the way this works, you've got a tRNA coming in, it's parking in this site. Actually, this is the E site, because what just happened here? What just happened right here, you think? This tRNA had already delivered his passenger. He doesn't have an amino acid up here. He already let it go. What's he doing now? He's leaving. He's going to look for another passenger. Let me back up a slide or two here. Remember, the 3 sites in that large part of the ribosome are called the A site, the P site, and the E site. The E site is where it leaves. It's where it exits. tRNA goes, pulls away, pulls out of the parking lot. The P site, the A site, is where it actually first comes in and parks. The P site is where the protein is getting built. It's where what's getting attached. One amino acid to the next. So let's go back here for a minute. Here we can see, well, I wish we'd seen this picture better. Let's look at it here. This one's getting ready to leave, but this tRNA is bringing in his own amino acid, phenylalanine. PHE is an amino acid. What do we say these three letters right here are called? That's a codon. That's on the mRNA. The only tRNA that can park there is a tRNA with a complementary code. AAA, the only thing that'll go with that in mRNA is what? UU. So that code, it's got the password. He comes in and parks. The only passenger that particular tRNA will ever carry is what? We said to think of these tRNAs like a racist taxi cab drivers. They'll only carry a particular what? A particular passenger, a particular amino acid. The only amino acid this guy will ever carry is that one. Okay, so that ensures that the right amino acid is put in the right spot. So he comes in and he parks in this A site, and when he does, what's going to happen between valine and phenylalanine when he parks in here? When he parks, these two amino acids are going to get bound together. They're going to make a bind. And what did we say we call that bind between amino acids? Call it a peptide bind. Takes a little energy, and what did we say is going to provide the energy to make that happen? GTP. All right, and then the last thing we get to is what's called termination. And go ahead and notice from the picture that in termination, everything comes apart. The mRNA comes loose from the ribosomes. What would this be right here? What is that? That's a polypeptide chain. That comes loose. The small subunit of the ribosome and a large subunit of the ribosome disassemble. And what tells them to do that? Yeah, eventually they get to a certain code on the mRNA that says what? Stop. You're done. Polypeptide chain is finished. Stop here. Now there's that actual code sequence that we'll see in the table here in just a second. So that last step is called termination. You're stopping. This occurs when a termination codon reaches the A site of the large subunit. And it basically says, stop. When it does, a release factor, an enzyme, comes into that site, does a little hydrolysis. What's hydrolysis mean? Splitting stuff, using water to split stuff. It splits apart the ribosomes and the tRNAs and the mRNAs, and everything separates. And the protein is now finished, or at least the polypeptide is finished. What do you think happens to that mRNA? Is it done? That same mRNA can be what over and over again? Can be translated over and over again to make that same protein many, many, many times. And we're done from there. Questions? Remember, the A site is where what happens? That's where the taxi parks. He brings in his passenger. The P site is where what happens? They get attached. They hold hands. They make a what kind of bind? They P site and peptide bind. That's where that happens. And then it doesn't show it in this diagram, but the E site is where the taxi cab what leaves. Right. Let's end with this. This is what our little lab activity is going to be about. Let me get a blank slide so we can see what we're doing here. All right, let me get out here to the end. That was great. Try that again. All right, let's say that I gave you this sequence of DNA. Let's say I told you we had a piece of DNA that was 3 prime, and that its codons were this. Let's say it had a T, TAC, CCC, A, GGC. And eventually, let's say it also had a ATT, and it ended at 5 prime. So this is a piece of DNA. Now, is that a template strand of DNA or the other strand? We're going to read these left to right just like we would a book. So this would be the template or the non-template piece of DNA. Which one? It's the template. Template DNA always runs from 3 to 5 because RNA has to be built from 5 to 3. So this is the template. Let's say that I wanted you to tell me from that template, I wanted you to tell me the sequence for the other strand of DNA, the non-template strand. Well, everybody in here can do that, right? If this is 3, what's the other strand got to be? 5. Let's do that in a different color. It's going to be 5. And if it's TAC, we're going to get what? AUG. Not AUG. We're going to get ATG, right? We're talking about DNA. And we're going to get G, G, G. And we're going to get T, T, T. And we're going to get C, C, G. And we're going to get T, A, A. And that's going to end in 3 prime. Let's say I also ask you for the mRNA sequence that we would get from this. mRNA is made from which one of these strands? The template. If the template starts with 3, the mRNA has to start with? The template starts with 3, the mRNA starts with 5. All right, now remember, we're using this as the template, not this. This is the template. This is the thing we copy from. So if the template says TAC, our mRNA is going to say AUG. If our template says CCC, our mRNA is going to say GGG. If our template says AAA, our mRNA is going to say UUU. If our template says GGC, our mRNA is going to say CCG. And if our template says ATT, our mRNA is going to say UAA. And it's going to end in what? 3 prime. That's our mRNA sequence. Let's say I also ask you for the sequence of anticodons, which is what? tRNA. Now, I didn't show you a picture of tRNA. I need to do that here in a minute. Each tRNA is separate, right? This is one strand. The mRNA is one piece. But each tRNA only has how many unpaired bases on it? 3. So it's each set of 3 is a different thing. But if the mRNA says AUG, what's the first anticodon have to be? AUG is a codon. What's the anticodon have to be? It's RNA. UAC, right? Codons have to be complementary to what? Anticodons. And it's RNA, so no T's. So if this is GGG, this has to be CCC. If this is UUU, this has to be AAA. If this is CCG, this has to be GGC. And if this is UAA, this has to be AUU. All right, if somebody with a sheet of paper, Megan, if you would write down this mRNA code for me, just on a sheet of paper, because we're going to go to another slide here for a second. Let's say I also asked you for the sequence of amino acids, the amino acid sequence. Well, it turns out scientists have figured out what all these codes mean. They've decoded the code. They know what AUG codes for. They know what GGG codes for. And there's a table in your book on page 314. If you got your book, get it out and look at it. Actually, I think I'll just put it on the Elmo here for a minute. Now, unless you're told otherwise, this is called the genetic code table. Unless you're told otherwise, these genetic code tables are usually based on what strand? The mRNA. Now, in the little lab activity we're going to do in a minute, it specifically tells you that table is based on tRNA. But almost always, unless you're told otherwise, when you're using one of these tables, it's an mRNA table. So if we go back to if we go back to our strand, our first codon, our first mRNA codon, Megan, was AUG, right? So we go to this table. Notice over here's the first base, up here's the second one, and over here's the third one. So we go to A, that tells us this row. We go to U, that tells us this column. And then we go to G, that tells us right there, AUG. And that codes for this amino acid, MET, which we learned a minute ago was what? Methionine. So the first amino acid in our chain, we're just going to write down the abbreviations. Methionine. What was our next codon? GGG. So G row, G column, right there, GGG. That stands for glycine. You don't need to know what these abbreviations stand for, just know the abbreviation. GLY. That's our second amino acid. Those get hooked together by what kind of bond? Peptide bond. Ribosome reads over. What's the next codon it reads? U. So U row, U column, right there, PHE. That's our third amino acid, and that's phenylalanine. That one gets bonded to glycine. Then we scoot over and we read CCG. So C row, C column, G right there. That's the amino acid proline, PRO. That gets bonded to phenylalanine. Scoot over and we get the what? UAA. So UAA. And that puts us right there, and that says what? Stop. That's the stop codon, termination codon. That tells us translation is what? Finished. So we stop. We don't need to write down stop. Stop's not an amino acid. We just stop. This is now what? What is that? It's a polypeptide chain, and several of those are going to get wrapped together, and when they do, we're going to have what? A protein. Does that make sense? All right, let me get out of here for a minute.